Good evening, everyone. I'm really pleased with the nice turnout. I'd like to welcome you all here. My name is Ralph Cohen. I'm a faculty member in the mathematics department, and I'm the Senior Associate Dean for the Natural Sciences in H&S, the School of Humanities and Sciences. The discussion that you're about to hear and participate in is the second in a series of discussions about various topics in the natural sciences that we'll be sponsoring. These events will generally be panel discussions like the one we're having here tonight, where the panel will consist of some of Stanford's leaders in the area under discussion. The only goal of this series is to simply have a forum for groups of Stanford faculty to communicate to their colleagues across other scientific disciplines their excitement around current work and projected future development in various areas of the mathematical, physical, chemical, and biological sciences. Now, as many of you remember, the first of these discussions was held back in December. It was a fascinating discussion sponsored by the physics department about cosmology and the beginnings of the universe. This evening, we'll have another fascinating discussion, I'm sure, sponsored by the biology department, this time about evolution. So we've gone from the origins of the universe to the origin of species. Our panel this evening consists of some of Stanford's leading scientists studying evolution, so I'd like to introduce them to you. First, Professor Liz Hadley. Liz is one of the world's leading environmental biologists. She is the Paul S. and Billy Achilles Professor in Environmental Biology, and she's a senior fellow at the Woods Institute for the Environment. If that isn't enough, she's also the Senior Associate Vice Provost for Undergraduate Education. Liz and the members of her lab are involved in a wide variety of research projects, but they all share a common interest, namely what determines and maintains vertebrate diversity through space and time, and how is that diversity influenced by the environment. Next, we have Professor Dmitri Petrov, also of the biology department. Dmitri is the Kevin and Michelle Douglas Professor in the School of Humanities and Sciences, and he's also the Associate Chair of the department. Dmitri is one of the world's leading computational biologists. He and his lab do theoretical, computational, and experimental work to address questions in molecular evolution and molecular population genomics. I'm particularly pleased to introduce to you one of Stanford's newest faculty members, Assistant Professor Erin Mordecai. This is Erin's first year on the faculty of Stanford in the biology department. Her appointment was a product of an international search for a biologist specializing in ecology and evolution. As I understand it, the aim of her research is to investigate the effects of pathogens on plant diversity and the impact of climate change on the ecology of disease. We were really thrilled when Erin accepted the offer to join Stanford's faculty, and we're very pleased that she agreed to participate on the panel discussion this evening. Finally, I'd like to introduce the moderator for the discussion, Professor Tim Stearns, the chair of the biology department. Tim is the Frank Lee and Carol Hall Professor of Biology, and he's also a professor in the Department of Genetics in the School of Medicine. He's a member of BioX, and he's also a faculty fellow in the new ChemH, the Chemical Biology Institute. He's one of the country's leading molecular and cell biologists. So without further ado, let me hand this over to Tim, leading the panel discussion entitled "Back to the Future, Is the Course of Evolution Predictable?"
All right. Thank you, Ralph. I have my own microphone, and I'm going to use the board for a brief introduction to the topic. Now, actually, when I came in this room before we were setting up, I realized that I gave the first lecture in this room in 1997 in a course that my colleague James Nelson and I were teaching back then. Our picture was on the Stanford Daily's cover of teaching in this new room. And that I'd only been an assistant professor for a couple of years then, actually. And back then, when I was a young assistant professor, or even beforehand, I assumed that at universities like Stanford, scientists did this kind of thing all the time. They got together and they talked about the great ideas in their fields, and other people came to listen and all of that. And of course, I only realized after coming that things like grants and other stuff all interferes with that wonderful notion. And so I'm so thrilled that Ralph took the initiative to start this kind of event so that we can have these kind of discussions. And I really enjoyed the physics one that we had a few months back. Now, the title of this evening is Evolution Predictable? Now, that could mean many things. So it could mean, is it predictable that starting from the ground state of Earth back at the beginning, you would end up here in this room with people talking and listening to each other? You could view it as a question of, is it a given that in the universe, with the chemistry that we have in the universe, that one would end up with the kind of life that we know of? You could view it as a much more short-term question than that. And you could ask, is it predictable in the current day situation, if there's some perturbation to the environment, is it predictable how organisms will change? And you can even view it in a very short scale, which is actually very relevant to medicine, which is, for example, will this mole on my arm turn into a melanoma by genetic change, which is a form of evolution as well. And that's the kind of thing that happens over months and years. So all of these timescales are the kind of things that we're going to talk about tonight. And I want to start by sort of laying out the ground rules for understanding biology with respect to evolution. Now, some of the things I'm going to tell you are things that some of you know very well. Indeed, some of it was probably discovered by people in the audience. So forgive me for getting very basic, but I'm going to do it at the board. And I want to talk about four things. Okay? So the first is the timeline of the beginning of life. Okay? So in the beginning, way back, although not back all the way to where physics was talking about, I'm going to start back 4.5 billion years ago. Okay, so back then we had Earth. Okay, Earth was in the beginning a rather inhospitable place for life, but sometime after Earth formed, it became a warm and wet place, which we now believe to be a place where life could form of the sort that we know. About 3.5 billion years ago, the first cells, so the first life as we know it, let's say, forms. And really this is about 4 billion to 3.5 billion. I'm going to draw this as a representative of the kind of life form that existed back then. And it looks a lot like a current day bacterium, if you'll grant me the sort of size scale here. About 1 micron, let's say, is that. Okay, so about 1 micron wide on that scale. And this transition from no life to life is mysterious. We are not going to answer that today. Okay? There's a lot of hypotheses as to how you get this first cellular life form, okay, which looks a lot like present-day bacteria, but there's really no good answers to how you get there. And I have to say, this, although simple in respects, is already an incredibly complicated machine. So this last common ancestor from which all life as we know it came was already a very complex cellular organism that was able to replicate and do metabolism, et cetera. More time passes, and about 2 billion years ago, something that resembles eukaryotes, the kind of cells that we have and that we are, forms. I'm going to indicate that like this. And I'm going to say there's also the potential for many of them to get together. And I'm going to give you a size scale that's a little bit bigger. Let's say that's about 10 microns. Okay? And the reason I drew it like this is because we think that eukaryotes evolved by engulfing some other bacterial-like cells that gave this new cell distinct properties. Okay? So the eukaryotic cells that make up our bodies derived back from some initial event on the order of 2 billion years ago. Okay? After that, evolution happened, and everything that we know of as life today came from these things. So for the most part, if you look around you, all macroscopic life came from this event of the formation of eukaryotic cells, and much, although not all, of microscopic life came from these prokaryotic cells. Now, I drew it as a timeline, essentially. right? Sort of indicating that one came from another. And I said that everything that we know of now came from these two types, right? How do we know that? Well, we know that because if you look inside of these cells, everything works exactly the same way. So the fundamental notion that everything is the same with respect to life on Earth is, I think, perhaps the most important thing one can understand about biology. And in particular, the thing I want to mention to you that is important for our discussion, and again, everyone is familiar with the basic notion of this, is that back in the beginning, this first cell, it already had all of the bits for information transfer, that is, passing on traits from one generation to another. Okay? And you know that information in biology is contained in many different ways, but the chemical form of information that's passed from generation to generation is, of course, in this molecule DNA. It's a long polymer of relatively simple chemicals, and there's 4 of them, and it's the sequence of that that determines the information. That goes to RNA. This is the central dogma of molecular biology. And that goes to proteins. This is all figured out from the 40s to the 70s. And there's some variations on this. It turns out this isn't unidirectional. It can go this way too. But largely, the information goes from this polymer, this simple, long polymer of 4 different compounds, okay, to essentially a messenger between that, and then to make this complex macromolecule proteins, and that's what makes up most of our cells, okay? That's amazing, again, because if you think about it, how did this happen? Nobody knows. It requires the ability to make this polymer, reproducibly replicate it every time you're going to divide, and then to translate that information that's in this long string of letters into something three-dimensional, proteins, cells, organisms, okay? Absolutely phenomenal. It's the founding concept in molecular biology, that entire field. There's much to know here that we still don't understand, although again, the basic tenets of this were figured out now, back in the 60s and 70s. All right, so that's the evidence that everything is the same. Everything works exactly the same way. Is it necessarily true that, given the chemistry of life, it had to happen that way, and it would happen again if you provide a warm, wet planet with the suitable organic compounds? Unclear, okay? And I will, again, remind you that the first thing we know about is already massively complicated. Right? So we really don't know this first event. Okay. I also want to give you some numbers to remind you of the scale of information that we're talking about. So for these guys, okay, the genome size, that is the number of bases in this DNA, the number of positions, is about 5 times 10 to the 6th. Okay? For us, I'm a scientist, excuse me. Okay? It's about 3 times 10 to the 9th. And it's actually times 2 because we have 2 copies of our genome. All right? That's the scale of the information that is present in each of our cells. These number of bases makes on the order of 5,000 proteins. This makes about 20,000 proteins. That's all we have to work with for making our cells as organisms and to change in evolution. All right, so the beginning, everything's the same, but then the next important concept is that everything is different. Because you know that if you look around at the other people in the room, we're all different. And if you look at other organisms, of course, we're all different. So there's variation. Okay, so this notion of variation is critical. And I want to remind you that variation, ultimately, the ultimate source of variation is change in that information. All right, so change in the DNA, that is mutation, that's passed from generation to generation, provides the fodder for evolution to act on, this variation, okay? And to understand that, you have to understand maybe another number, which is the mutation rate. So the mutation rate, it differs in different organisms. So I'm going to give you a general statement about mutation rate, which is that it's about 10 to the minus 8 per base, that is position in the DNA, per generation, that is per cell division or every time you replicate the DNA. Okay? Now, from the chemical perspective, it's an amazingly high fidelity machine. Rarely makes mistakes. But from the perspective of an organism like us or from bacteria growing in the soil, there's a lot of cells in those situations. And so you have, in our bodies, there's on the order of 10 to the 13th cells. For a colony growing on a petri dish of bacteria, which some of you are familiar with, there's on the order of 10 to the 7th or 10 to the 8th cells. So mutation rate is low, but the number of cells is high, okay? So change happens. There's a lot of discussion, but no real understanding, that life had to have evolved this way. That is, it isn't perfect in transmitting the information from generation to generation, and that imperfect nature is what's important about life as we know it now, because that allows for variation. The presence of variation allows for evolution to happen. Okay? The last concept is that once you have variation and you have a population of individuals that differ from each other genetically, you have the opportunity for the fraction of individuals in that population that have, say, one version of the DNA versus another version to change. If they start out equal, one could go up, the other could go down. Now you end up with more individuals with this type than this type. Okay? That change is what the study of evolution is really all about. Okay? So understanding the factors that result in that change and how that plays out on the different timescales that I mentioned at the beginning is what this is really all about. Okay? And you all know Darwin's use of the term natural selection to describe one of the processes that results in changing the frequency of different individuals in a population, where the notion is that some do better than others based on their variation, and they will then come to predominate in a population. There's also artificial selection, which Darwin also uses as examples in his book. But there's other kinds of events that happen during evolution, too, that will change the fraction of individuals in a population. So all of these factors come into play when thinking about evolution as it's going to be discussed by the panelists today. Everybody good with that? Any questions?
Yeah.
Yes, sir.
How do you define the base there?
Okay, the base in this case refers to the base, it's the chemical that makes up DNA and RNA. So base, in this case, refers to, in the common parlance, A, C, G, or T. Right, so there's 4 bases that make up DNA and RNA, okay? So it's a chemical, it's the subunit of the polymer.
I understand it's a chemical, but how do you count it?
Oh, the way you count it is you can sequence the DNA. That is, it's a long polymer, and there are methods for determining the order of the bases in a long string of the DNA, okay? And these methods first method was in the 70s. We now have methods that do this extremely cheaply and extremely rapidly, and it's one of the things that has driven the revolution in understanding many things, including evolution. Good? Any other questions? Yes?
Where do viruses fit into this?
Oh, okay, so this is exactly the question. We were talking about this just before we started today, and the question was, should we mention viruses? Because they're always complicating. They're not cells. They are bits of information, nucleic acid, that is transferred from cell to cell and replicates inside cells. Is that living? I would argue no, but others might argue yes. And so I think we won't answer that question, okay? I'm sorry.
Could it predate bacteria?
Almost certainly not, in that viruses are usually bits of information that come from the cells that they infect, okay? So this is a chicken versus egg thing, so take it with that in mind.
But they do evolve.
They do indeed evolve. They evolve more rapidly, actually, than in most cases than cellular life forms do. Any other questions? All right, I'm going to allow my colleague Dmitri Petrov to have the floor.
Can you hear me? Well, thank you very much for coming. I want to thank Ralph again for organizing these evenings. I hope they'll become a tradition, and today will be a success, and we'll have many more of these. So I will start the conversation about whether evolution is predictable. And so my job is to tell you some speculations and some facts of the matter and prime the conversation. I promise, even though the question is very big, I'll come up at the end with a firm answer. So we can't start talking about evolution without mentioning Darwin. And so this is the only illustration in The Origin of Species where Darwin imagines evolution as a tree, as descent with modification. Each lineage here, each line here is a lineage of descent from one organism to the other, with some branches through speciation coming off, some dying off, and continuing. And so time goes this way. And we all think of Darwin as coming up with a theory of natural selection that has this notion of necessity behind it, things happening for a reason and organisms evolving to adapt to the environment. But he himself was very aware of chance and talked about it in The Origin. And there are many reasons for why evolution could be stochastic and full of chance events. One of them, for example, is that organisms, they evolve, they change the environment, and therefore the process of evolution has a direction. It's not necessarily reversible. So for example, when organisms evolve photosynthesis and start putting out oxygen to the environment, they eventually change the planet, so now we have oxygen, and so the organisms that use oxygen now would not have been able to be there 3 billion years ago, for example. So since Darwin, we filled in a lot of the facts of the matter. We have accepted the idea of evolution and natural selection. I also learned a little bit about how organisms really have evolved and put some dates on some of the key events. And so this is a tree of tetrapods. And we know something about the time when the key splits have happened. So the fact has been established. But again, we're going back to the topic of today's discussion about the predictability of evolution. And the question is, do we necessarily have to have these organisms rather than others around us today or at any time in the past? And this question was put forth maybe most eloquently or most influentially by Harvard paleontologist Stephen Jay Gould, who also wrote a lot of popular books on evolution. And he had this idea of having this thought experiment of replaying the tape of life. So imagine you could go back in time to, we erased the Earth, beautifully drawn by Tim. So Earth 4 billion years ago, if we go back to 4 billion years ago and replay the tape of evolution, what are we going to see? So this good thought experiment. And Stephen Jay Gould was firmly on the side of chance. So he used this metaphor to argue that obviously we will not get what we have today. And as an example, he said, what are the chances that I will be writing this book right now, being a Harvard paleontologist, Stephen Jay Gould? And if you think about that, the chances are pretty low. There are a lot of chance events that had to happen for him to be sitting in that faculty chair writing that book. And it seems a very convincing argument that it's all chance. Then you start thinking a little bit beyond what he said, and he's a very eloquent writer, so very convincing at first. So I'm thinking, well, maybe Gould would not be there, but what are the odds of there being a paleontologist at Harvard? So if there is Harvard, maybe there is a bigger chance of there being a paleontologist of some kind, but maybe not necessarily Gould himself. Is there a chance of Harvard being there to begin with? Well, some people think that it's a very unlikely event. But you know, if there are universities and there being Boston, maybe the fact that there's a university there that's named after somebody is not so unlikely. What are the odds of there being universities, or self-aware primates, or conscious organisms, or life, right? So we can go all the way down and so ask the question, what do we mean when we ask, is evolution predictable? At what level of inquiry do we ask this question, and what level of inquiry is reasonable to ask this question at? So we can ask the same question when we look at organisms around us. We can ask, when we see evolution, is it likely to happen through specific mutations of specific genes? Is that the level? Or are we interested in ecological roles that organisms have, for example, predators and prey, maybe specific phenotypes, or key innovations, or outcomes, but not necessarily the paths, the way you get there, or statistical properties of evolutionary process, maybe the distribution of organisms or distribution of body sizes, or the levels of diversity, right? So I'm putting up all of these questions just to, again, to prime the conversation, to maybe ask you, what do you want to know about evolution? How would you like to predict it? So we cannot go back in time 4 billion years ago, but in a very nice way, there have been natural experiments of playing the tape of life many times over again. And I'll tell you about some of them. So one of the most profound ones is the parallel evolution that happened in Australia, South America, and the rest of the world. And I just couldn't help but, let's see, I'm just going to play. So this is a little movie. This is the movement of tectonic plates. I'll go a little slower. So the counter there is the millions of years ago. So the tectonic plates are moving around. Eventually, they're all coalescing. And then something like 120 million years ago, you can see South America coming apart from Africa and spending about 100 million years in what was termed splendid isolation. And the same is happening to Australia. Eventually, South America hitting North America 3 million years ago, creating Central America, and organisms start mixing with each other. But we have fossil record of the organisms that lived in South America for this 100 million years, and we have encountered them in Australia to a large extent. And what's amazing is that this set up an experiment, especially when they came to mammals, because at that time, the South America and Australia primarily were populated by marsupial mammals, and they are different from eutherian mammals or placental mammals that we're more familiar with. And so there we can ask, so we had two independent evolutionary processes of eutherian and marsupial mammals. Do we end up having similar organisms in terms of the way they look or in terms of their roles? And so these are the ones that we familiar with from placental mammals. And amazingly, at least to me, if we look at the marsupial mammals, there are a lot of organisms that look very much the same. And the same to the extent that's a little spooky, not only in terms of their roles, but also just in terms of their morphology. The one that always strikes me as the most remarkable is a saber-toothed cat. That there was a marsupial saber-toothed cat and the eutherian saber-toothed cat. So this is marsupial one, this eutherian one, and they basically look the same. Now, if you had asked me what are the odds of evolution, if you really play tape of life, you would end up with a saber-toothed cat that was going to look like that. I would say the odds are pretty low. And the fact that it happened twice independently says that there was some kind of a necessity of that kind of an organism arriving through evolutionary process, maybe in the course of a long period of time, but eventually arriving. All right, so that's one experiment. So let me ask you, you can say, okay, so saber-toothed tiger, it's good to have big teeth, and seems like a good idea. So maybe whenever you think of some kind of a great idea, it needs to happen many times. So I'm just going to ask you to predict how many times, or tell me, how many times certain features, certain innovations evolved. So flight and animals, how many times has it evolved? Once, twice, more than twice? At least three, many. Great. So insects, birds, bats, right, and others. So here's another one. Plants need nitrogen, right? We all know that we need nitrogen. We need fertilizer for plants. There's a lot of nitrogen in the air, molecular nitrogen. So how many times did plants evolve ability to fix molecular nitrogen from the air? What do you think? Seems like a good idea.
By themselves.
What?
By themselves.
By themselves. They just, so you can take the nitrogen from the air and just incorporate it into nitrogenous compounds that the plants need. What do you think? If I were a plant, I would like to be able to do it. It's kind of silly to be sitting there awash in nitrogen and not being able to get it. Never. So they know how to do it through bacteria. They have this ridiculous way of associating with bacteria and getting bacteria to do it for them. So we know it's possible to do, but they never have been able to do it independently of bacteria. So why, I don't know. So this is just in terms of, again, predictability. Eyes. So the eyes is often used as an example of something very complicated, very hard to put together, and impossible to evolve from scratch. So how many times did eyes evolve? 2, 3. So people count, so some people say 40 times. Right, so amazingly, the eyes have evolved many times independently, even though they actually use very similar internal machinery. They use the same opsins. The light-catching machinery is the same, but eyes as a structure evolved many times. So I think that, when I read it, I did not have an intuition that that would happen many times. So, all right. So a few more examples, and I'll be done. So here, there are more cases where we have these cases of replaying the tape of life. One of the study systems that was worked out at Stanford and by many people, but particularly at Stanford and David Kingsley's lab, is a case of little fish called three-spine sticklebacks that repeatedly live in the marine environments, but repeatedly go into the freshwater ponds. And this freshwater pond, they go into the ponds many times, and so every pond ends up having an independent evolution of the marine stickleback into some form that's more adapted to the freshwater. And repeatedly, what happens, the sticklebacks in the marine form, they're covered with plates, and they have this very big pelvic spine that help when they're three-spine sticklebacks, they go like this, and it's hard to swallow them. The freshwater ones lose this structure repeatedly in all of the lakes, right? So it's possible to find out how they lose it, and they lose it through one particular, recurrently through mutations on a particular gene, regulatory structure of a gene called PTX1. So that's already quite remarkable because in different ponds, the same mutation, the same gene happens. What's even more amazing to me is that the same kind of a process of loss of pelvic structures, in this particular case of hind limbs, happen independently in whales and cetaceans and in manatees. What's crazy is that it looks like in these cases, there's some evidence that in all of these cases, the change happened through the mutations in the same gene again called PTX1, suggesting that in these very diverse organisms, they're vertebrates, if you want to lose hind limbs, you're going to be doing it through one particular genetic pathway. We can also do play the tape of life in the lab. So now we can just take, it's easiest to do microorganisms. You can do it with flies or C. elegans or worms. You can take E. coli, put it in a flask, and propagate it many times independently. And every time you propagate it, you have an independent tape of evolution. And you can see what happens. So I'll just tell you very briefly, there was this experiment that I love, published in Science 2011, where Olivier Tenaillon set up 115 of these lines, let them go for a few thousand generations, and they were evolving ability to grow at 42 degrees, which is something that they don't like to do. They all got to grow much faster by the end, and then he looked to see where the mutations were in the genome of these guys. And each line here is the genome of E. coli. These are different mutations, and you can see there is a. I'm not asking you to look at particular ones, but overall the pattern is that there's a lot of differences. There are some places that get hit over and over again. Right? So there is both necessity and chance if you look at it at that level. And this is the graph that shows the probability that any two of these strains at the end share the same mutation. And the probability is very low. That the mutations happen in the same place of the gene is also low. But then if you look to see which genes are hit in independent lines, they tend to be much more likely to be the same. If you ask which groups of genes that act together, higher yet. And if you ask which functional units, it gets even higher. So again, you need to ask the question, at what level do you want to know what's happening in the system? Very similarly, I was lucky to be part of an experiment done in Gavin Sherlock's lab in the genetics department using this technology that was developed by Sasha Levy, where we can barcode individual genomes and detect adaptation occurring in the flask in yeast as it's happening. And so this graph, I'm not going to go into the details, but this graph, this red line show the trajectory of adaptive events happening in the genome somewhere in this population. And there are a lot of them. There are like 25,000 adaptive mutations sweeping this population at the same time. What's amazing to me is that this is one flask. So these are generations, and this is the number of cells on logarithmic scale, 1,000, 10,000, so forth. So this is how it's happening in one flask. When we do it in a different flask, it looks more or less identical. So when you look to see how evolution happens in the detail, it happened more or less the same way. The way the whole population gets better is here. This is the fitness increase, almost identical. And when you look at the detail, the similar genes are hit, but not in the same place. So again, some things are very predictable, and some things are not predictable at all. The final thing I want to tell you is that we might want to know this not just because it's kind of cool and interesting, but also because these are exactly the processes that we often deal with for entirely practical reasons. So viruses were mentioned. Viral infections are a big problem, and viruses exist in very large populations and have very large number of mutations. And it turns out that the evolutionary course that viruses take are often predictable. In case of HIV, evolution of the virus inside your body is what kills you. So you get viral infection, you get HIV, you have a 2-week bout of flu-like symptoms, but then the virus stays in the body and continues to evolve, eventually decimating the immune system. And it evolves through adaptation to the immune system. And it turns out that course is entirely predictable. The early drugs that people came up with turn out not to work. They slowed down the progression of HIV, but by a little bit. So this shows the progression of how the immune system declines. So this is in HIV. This is if you give one particular drug called AZT. It slows it down by about a year, but predictably the virus gets resistance every single time, more or less. Turns out now that we have figured it out, and we have combinations of drugs that work, the resistance more or less doesn't evolve, or evolves in very few percent cases. And that happens if we give virus more complicated tasks, giving complicated mixtures of drugs. So this is a case where we care to stop adaptation from happening, and we know when it doesn't work, and we know when it does work to stop it. And so it's a good example of something we need to know how to do. Cancer is another big example. I'm not going to talk about it, but cancer is an evolutionary process, again, happening recurrently in individuals, resistance to chemotherapy happening recurrently as well. So just to finish, I promised you a firm answer. Is evolution predictable? My answer is yes. It's a firm answer, sort of. Right? And the question is, can we predict it in a way that matters to us? And I think in many cases we can. So I think that we should be cautiously optimistic.
Okay. Thanks, Tim and Dmitri, so far. Really interesting discussion. I'm going to move now into a much more specific application of this question. When can we predict evolution? And so I'm actually not an evolutionary biologist, but I study disease ecology, and I study diseases like malaria. And a lot of times we want to know how diseases like malaria will respond to future changes in selection. So that's the example I'm going to talk about today, but it's sort of one example of this broader idea of when can we predict and how can we predict when things will evolve. So I'm going to address this question: will malaria transmission adapt to warmer temperatures if the climate changes in the future? Okay, So malaria transmission, rather than being a single species problem, it's actually a multi-species problem. So many of you are probably already familiar with this, but malaria is a parasitic disease. It infects 250 million people every year. And to get infected with malaria, you have to be unlucky enough to be bitten by an Anopheles mosquito that's carrying the parasite. And not only is the mosquito carrying the parasite, but the parasite has to have gone under a development process within the mosquito to be ready to be transmitted. So the malaria transmission cycle basically requires a mosquito biting a person that has the parasite, and then the parasite develops in that mosquito over a course of time ranging from 3 days to several weeks. And then after that development time, that mosquito will go on to infect every human that it bites after that, or at least most of them. So as you can imagine, this malaria transmission cycle really depends strongly on what's going on in the mosquito. And actually, as you probably know, we've been studying malaria for a long time, over a century, and we've done a pretty good job of figuring out what processes control transmission. So these are things like mosquito biting rate. So how often are these mosquitoes biting? How long are they surviving? How capable are they of transmitting the parasite? How quickly does the parasite develop within the mosquito? And how quickly do humans recover when they're infected? Now you might imagine that because these things are all taking place within the body of a small-bodied, cold-blooded insect, that they're susceptible to temperature, or that they depend on temperature. We all really know this because we think of malaria as sort of a tropical disease. We kind of think of it as requiring warmer temperatures. And because of this long history of research in this system, we can actually predict pretty well how transmission responds to these different mosquito and parasite processes. And all I want you to get from this equation here is just that R0 is a metric of transmission into a susceptible population, and it depends non-linearly on all these parasite and mosquito traits. And all of these traits, like I said before, are temperature sensitive because they take place within this mosquito. So it turns out to predict how malaria transmission responds to temperature, we really just need to understand the physiological responses of the mosquito and the parasite to temperature. And we're actually pretty good at doing this. So here's one example of a vital rate that affects transmission. This is mosquito development rate. And we see this really characteristic hump-shaped response to temperature. We see this across all sorts of taxa and all sorts of traits. And it's both theoretically and empirically predicted. So it's not at all surprising that we see this relationship. So in this case, mosquito development goes to 0 around 16 degrees Celsius, and again around 34 degrees Celsius, and it's peaking somewhere in the middle, around 28 Celsius. So we can look at all of those vital rates that I mentioned earlier that drive transmission and see how they respond to temperature. And in fact, again, we see this really predictable pattern of hump-shaped thermal responses. And in fact, just by measuring these vital rates in the lab at different temperatures, we get a pretty close correspondence with what models would predict. So now, just by knowing about these physiological responses to temperature, we can combine those into that nonlinear model of transmission that I mentioned earlier, and we get a prediction of how transmission responds to temperature. So here's our prediction. It increases from 0 around 16 Celsius, peaks around 25, and then goes back to 0 around 34 degrees Celsius. And when we look at transmission in the field, what we see more or less corresponds to this prediction. So we see the highest levels of transmission going on around 25 degrees Celsius, and we see transmission really steeply declining above 28. Now, of course, there's lots of other variation that you can see here, which is not due to temperature. It's due to things like having bed nets, or having screens on your windows, or having more or less moisture in the air. But nonetheless, we can get a pretty good idea of how temperature drives transmission by comparing this mechanistic prediction with what we see in the field. So, in case some of you aren't familiar with Celsius versus Fahrenheit, 25 degrees is only about 77 Fahrenheit. So it's much cooler than you might expect if you think of malaria as a tropical disease. So basically what this suggests is that with future temperature warming, we'll have some places that are sort of at the cool margins of transmission, and as temperature increases, we may see transmission really increasing in these places. But the places that already have the most transmission are already at or above that 25-degree optimum. And so we actually expect in some of these places that warming might actually slow down transmission of malaria. Now, this is a really important prediction because it suggests that the places where malaria is currently the biggest problem may actually get better and not worse, to the extent that temperature is driving transmission. So the question you're probably all thinking now, since we're talking about whether evolution is predictable, is are we really going to see this decline in transmission? Now here's what the expected decline would look like if we mapped it onto Africa, which is where a lot of malaria transmission occurs. So we have a current hotspot in West Africa, sort of the Gold Coast region. This is an area where temperature is really suitable for transmission, and there's a really high population density. So there's a lot of people exposed to malaria right there. Now, over the next several decades, we're seeing this transmission hotspot shifting from West Africa to the Albertine Rift region in East Africa. This is somewhere where it's currently just a little bit too cold for transmission to be optimal. So with future warming, we may expect that transmission to become more optimal. Now, the other important thing to notice here, I haven't figured this out yet. There we go. This Gold Coast region here is no longer very optimal for transmission. And that's because it's actually becoming too warm for transmission to be optimal. It's becoming higher than 25 degrees a lot of the time. So this is our prediction. Will this actually happen? Will malaria be able to adapt to this future warming? And actually, will transmission increase at warmer temperatures in the future? I'll tell you already, I don't have a nice firm conclusion like Dmitri did. But I'll do my best. So basically what we want to know is, are these thermal responses going to be able to shift? So these critical mosquito and parasite vital rates that drive transmission, will they be able to have optima that increase with increasing selective pressure for warm temperatures? Well, I think there's some evidence in either direction. We don't know for sure, but we think that actually it may happen because there's already some local adaptation in different mosquito species. So mosquitoes that are more adapted to cool temperatures now. So for example, there's an Aedes mosquito species that is from Japan, and when it invades in North America and Hawaii, we see some evidence of local adaptation, that it might be doing better. There might be locally adapted populations that are doing better in warmer climates than the original parent population. So there's at least some evidence that local adaptation to temperature may occur. We also know from the evolution of resistance that these mosquitoes and parasites are really good at responding to selective pressure. So when we apply a bunch of insecticide or a bunch of drugs, these mosquitoes and parasites are very good at finding ways to get around those obstacles, which suggests in general that evolution can happen in malaria. We also know just from the numbers game that there's a high probability that some parasite out there probably already carries an adaptation that would be advantageous in warmer temperatures. So any individual human infection, if I get infected, my infection will have 10 to the 12th malaria parasites. And it'll be seeded not just from one parasite genotype, but from multiple genotypes, because a lot of mosquitoes carry multiple parasites. So already you have enough abundance of parasites in an individual person that you could cover every single possible point mutation within the genome of Plasmodium, within my own infection. So that suggests that if you multiply that 10 to the 12th parasites per person times the 250 million people a year that get infected, you have a lot of parasites. You have 10 to the 20th parasites on Earth in any given year. So that just suggests that there's a lot of opportunity for these mutations to occur. And if they did occur and they were advantageous for surviving in higher temperatures, we would likely see them being selected. So okay, I think there's some pretty substantial evidence that selection will be strong and that we may be able to see some warmer adaptation in these mosquitoes and parasites. There's some evidence that that may not happen for these particular species. So, and I mentioned before that I hope I mentioned before that it's Anopheles mosquitoes that are required for transmission, and it's Plasmodium parasites. And there are 4 human Plasmodium parasites that infect us. So it's not like we have this huge diversity of malaria species. We have a limited diversity. And within these mosquito species, actually, the 25-degree optimum is pretty consistent across a lot of different dipteran species, different flies, as well as other insects and other predators. We actually see 25-degree optima pretty commonly. Now, whether this is because a lot of the world sees 25-degree temperatures a lot or not is in question, but you know, it's at least somewhat conserved that 25 degrees is the optimum for many organisms. We also know that organisms that live in the tropics tend to have narrower thermal ranges. So if you live in a temperate environment like we live here, we experience big fluctuations in temperature daily and over the course of seasons. So we all have to be able to tolerate many different temperatures. In the tropics, you don't see those big fluctuations. And so a lot of organisms are really tightly adapted to their thermal conditions in the tropics. So So that suggests that being a more or less tropical disease, malaria may have a, the mosquitoes and parasites involved in malaria may have a more narrow thermal range, which may limit its ability to expand at the upper end. We also know that the degradation of these processes that happens at the upper end is often sort of, if you go back to some of those asymmetrical curves that I showed earlier, things ramp up very slowly with temperature from cold temperatures, and then they drop off very steeply. So that drop-off can be very steep, and it can be limited by sort of denaturing of proteins. So there's some evidence to suggest that that upper boundary may be more firm than the lower boundary. So I've been talking right now about whether these particular mosquitoes and these particular parasites will adapt to temperature, these particular thermal responses. What about the process of malaria transmission as a whole? Will we see malaria transmission adapting to warmer temperatures in the future? Will we actually see this sort of fading out of malaria transmission in West Africa? I would argue probably not. Again, we don't have 100% evidence for this, but we have some good evidence that malaria in some form will probably persist in the future in warmer temperatures. One piece of evidence is that we know that there can be some vector switching. So Plasmodium may prefer Anopheles gambii. It may have the fastest transmission right now in Anopheles gambii. But we know that in the Sahel, for example, where we have this long drought period every year, there's another species of Anopheles that can sort of carry over transmission in between the period where Anopheles gambii dies out. So what happens is during the peak of the malaria transmission season, Anopheles gambii really drives transmission. But then in the dry season, there's another mosquito species that's able to estivate, or basically hunker down during the dry season. There's been evidence that individual mosquitoes can actually survive 5 or 7 months in these dry conditions. They just sort of hide somewhere, and then when it rains, the mosquitoes come back out, and very quickly you have malaria transmission again. So in this case, there's this ecological role for a second Anopheles species that's maintaining transmission. And it may even be that climate selected for this speciation event between these two Anopheles species. And both Anopheles are able to carry the Plasmodium parasite. So that suggests that even if the favored vector now were to blink out, there may be other vector species that could sort of come in and fill that ecological role. Now similarly, you could also have other Plasmodium species that could fill the role. So right now, Plasmodium falciparum is the most common and most deadly of the malaria species that infects a lot of people. But we've already seen some evidence that some malaria species from other animals can actually spill over into humans and infect humans and be much more devastating even than the human malaria parasites. So, for example, in Malaysian Borneo, we've had more and more encounters between people and the rainforest, and the mosquitoes that live in the rainforest, and the mosquitoes that live in the rainforest that also bite macaques. Those macaques have their own malaria species, and that macaque malaria species can actually get transmitted to humans. And it's now one of the major strains of malaria that's circulating in Malaysian Borneo. And it's very, very dangerous. A lot of people die from it. It's more deadly than any of the other human malaria species. So when there's the ecological opportunity, we see this switching of parasites. So in this case, it's a monkey malaria species. But in fact, there's actually over 100 different Plasmodium species that can infect birds, reptiles, primates, rodents, all sorts of mammals. So there are many Plasmodium species out there. So even if the 4 that are currently most common in humans were to blink out, there's a high probability that some other Plasmodium species would be able to spill over and start infecting humans and start transmitting within humans. So my broad conclusion here is that I think that it's an open question whether malaria as we know it now will adapt to future warmer temperatures. I think it's a much less open question whether malaria in some form will adapt to warmer temperatures. And in fact, it's even worse if we have some vector switching, some host switching of parasites, because that just changes the game. We understand what we know now about Plasmodium falciparum and Anopheles gambii, but as different players come into play, it's an even more challenging public health problem. So I think that this is both a really important and a really unresolved question, is how predictable is the evolution of malaria? And with that, I'm going to hand it off to Liz.
So it is so great to see this audience. I heard a little one back there, and we just have people, I think, from the community and from lots of departments and students, and it's wonderful to be standing up here. It's really exciting. So thank you to Ralph and thank you to Tim for hosting this. So what I want to do is take a little bit of a different perspective, and that is a perspective of deeper time. So thank you to Tim for going back 4 billion years. I'm not going to quite go back that far, but what I want to do is talk about the predictability of evolution over time. So the products of evolution through long periods of time. And I'm going to start with the fact that if you look at the past 500 or so billion years, the diversity of life on the planet has increased. So this is a record from the ocean, and these happen to be genera. If you remember, it's species, genus, kind of family. These are genera. The same is true when you look at the family level. It's much more difficult at the species level. And there are lots of versions of this. This was famously done by Jack Sapkowski at the University of Chicago. And this is just a track of the history of the diversity of life in the sea from 540 million years ago to the present. And one of the most amazing and still somewhat unanswered questions in the history of life on Earth is, why has diversity increased? There's lots of potential explanations. For example, there is something called the pole of the recent, in that the most recent rocks happen to be the most abundant and the easiest to find. They haven't been swept under continents. They haven't been melted away. And so the further back in time you go, the fewer rocks there are of the record. But even when you account for those things, there is still this very profound evidence that life seems to be increasing in diversity on the planet. The next thing that's important to know is that there have been some big events in the history of life on Earth. So there's this characteristic rise of diversity that happens at the end of the Cambrian, so called the Cambrian explosion. There's this extinction event here, the Ordovician-Silurian. There's the late Devonian extinction. There's the mother of all extinctions, the Permo-Triassic extinction, where at least 90% of the species on the planet disappeared. That is unbelievable. There is the the Triassic-Jurassic extinction and the Cretaceous-Tertiary extinction, when we lost dinosaurs and flying reptiles. Now, the key thing about these events is, in a way, they were restarting the evolutionary clock. To lose 90% of the species on the planet means that the diversity of life has to reassemble and re-evolve. And one of the key features of this is that it takes a long time. So on average, recovery from these extinction events is somewhere between 5 and 10 million years, although in some cases, in particular, this big Permo-Triassic event, it could have taken as long as 30 to 40 million years. So recovery from these extinction events, which takes place at a rate that at some point really is underlain by the mutation rate that Tim described to you, this kind of diversity is a diversity that accumulates through these ratchet events, where once you have a species divergence event, once you have the emergence of 2 distinct species from what was one, you can't go back to that single species. So this is the accumulation of novel species. It's not the species that transmit their DNA or that information horizontally, which happens in bacteria and viruses, as Tim described. By the way, I loved having my Drosophila molecular biologist introduce plate tectonics and paleontology to you. It's really fun to listen to him talk about that. So in terms of macroevolutionary diversity, I've very much oversimplified this, but here are the key 3 features that add to diversity over the evolutionary record. One is that, in general, the longer time you have, the more diversity will accumulate. And that simply happens because of this mutation rate Tim described, and it just happens that over time you accumulate more and more diversity. You add things like area, and this is either geographic area on the land, area under the ocean, area along some sort of a gradient, it doesn't matter. The more space there is for some kind of genotype, the more likely you are to get more and more genotypes. And then there's isolation. Isolation is a little bit of a mixed bag. In general, when you take two areas, they're evolving, they're on their own independent trajectory. By themselves, they'll accumulate similar amounts. If you have two same areas, two same times, they'll accumulate similar amounts of diversity. However, as Dmitri beautifully talked about, that diversity is in fact different. If you were then to add that back up together again, your diversity is higher. But from the perspective of either one of those areas, it's about the same. So isolation kind of goes both ways. Okay. So now this area thing, this is one of the, biology doesn't have a lot of laws. We're not like physics, okay? But one of the key laws, the fundamental laws in the science of diversity in biology is that in general, the more area you have, the more diversity you'll accumulate. This is, these are different continents, and these are subsets of areas in these different continents. And if you'll see, these are amphibians, these are birds, these are mammals. What we have here is just the number of endemic animals. These are log transformed, and then the size of the area you're looking at. And in general, all of these continents and all of these taxa show that as you increase area, you'll increase the number of species in that area. Now, these are continental patterns. And I'm going to tell you that in all these cases, if you add these up together, your global diversity is way up here. It's off this accumulation curve. The reason for that is because of this isolation idea. It's something we call provinciality. So each one of these continents have their own trajectory of evolution. They're accumulating species, and in some cases, they're moving species around. But it's key that the reason we have the global diversity we have is because each one of these continents has an independent evolutionary story. It comes back to bite us in a way, because what we're doing now on the planet, which is how I'm going to end, is essentially the prediction, our evolutionary predictions of the planet, is we're homogenizing the faunas from these different continents. We are moving organisms around. And the only fate, when you take a global diversity that's up here, that difference, these slopes are very, very repeatable. When you take the difference of the global biodiversity up here and you start homogenizing and mixing these faunas, you are going to decrease diversity overall. So we're losing area, and on the planet, we're losing area because of human population growth. Just quite frankly, that's the big reason. We are transforming ecosystems. We're losing intact areas for species to live. We do hunt animals. That's another reason we kind of deliberately are removing them from the environment. And then our climates are changing. And this plays a role. It's starting to play a role. It's going to play a huge role in the future in thinking about the diversity of life on the planet. Humans are pretty good at colonizing the world. This is a flow diagram of our out of Africa, basically, and we colonized Australia, we colonized the Americas last, we colonized Europe, and with the expansion of humans came extinction. Most of this is because probably, the jury's still kind of out on this, most of it is because we're pretty good and pretty efficient at hunting, especially when animals are relatively naive to us as predators. This is a menagerie of all the animals North America lost about 12,000 years ago. We lost giant ground sloths, mammoths and mastodons, these things called glyptodons, giant armadillos. We lost direwolves, saber-toothed cats. By the way, I want to interject, Dmitri said there were 2 saber-toothed cats. There actually have been 4 saber-toothed cats in the history of life. Smilodon, which is our California state fossil, A nimravid, which lived a long time ago, is neither a cat really nor a dog, but basically had a morphotype of a saber-toothed cat. And then two marsupial saber-toothed cats, Thylaco leo and Thylaco smilis, which occupied Australia and South America. Anyway, we had one in California right here, quite an extraordinary animal. Anteaters, giant bison, and camelids. Camels evolved in North America, migrated to Asia and South America, and then went extinct in North America. And now we know of them as the guanacos and the camels of the old world as well. So humans are pretty good at hunting, but we also transformed the environment as well. And that habitat loss likely contributed some to the demise of some of these animals. Now our populations are, in fact, dominating the planet. And what I'm going to show to you are estimates of biomass that I actually calculated for, or that have been calculated for humans. Since the Industrial Revolution, we have dominated the planet with our biomass. And in fact, ironically, these large animals we lost at the end of the Pleistocene around the world from between around 40,000 years ago to the present as we colonized the globe happen to be what we call the megafauna. So they're about 44 kilograms or larger. Guess what? We kind of fit in as megafauna. So we've replaced that biomass, not only with ourselves, but with our livestock and our pets. So the biomass on the planet now is actually dominated by human, livestock, and our commensal animals. And wild animals have hardly any biomass left. So if you just think about the biomass of living mammals, basically our occupancy of the planet is just unbelievable. This is with 7 billion of us. If fertility rates remain exactly the way they are now, the projection by the year 2100 is 27 billion people. There is no way we can sustain that many people. We can barely sustain the people we have on the planet today, and the only reason we're doing it is because we're mining the fossils from those past geologic time periods, 50 million years ago. And so clearly fertility rates are starting to drop, but they need to drop a lot more. This is a huge, huge issue. And humans, because we occupy space, we need resources, we need food, we cause extraordinary habitat loss. Now, what does that habitat loss do? So we basically modified 51% of our land area for our purposes. This is a map of cropland in the world. The only places we haven't completely transformed happen to be these either very remote areas, very inhospitable for cropland, or the places where there are biodiversity hotspots, in a sense where we have most of our species that are remaining left. So we now have dominated over half the planet and co-opted those resources, that energy, for our purposes alone. The other thing that's playing a role here is that all levels of biodiversity are at risk. This is a map of our protected areas on the planet. That's it. So in spite of not having completely transformed the Amazon, what's actually set aside for protection is small. Now remember, I told you that isolation plays a role. Isolation is a really big deal when the environments change, because species that say, well, let's pretend like your mosquito from Erin is going to need to move, or the mosquito host wants to move from here to here. I mean, isolation plays a big role. There's no continuous habitat that's protected for these animals to move. So as climates change, the continuity of their ability to kind of move where their environments exist will become harder and harder. But also, I told you that there's a strong relationship between biodiversity or species richness and the area they have to occupy. And if this is all the area we have set aside for biodiversity to occupy, out of what was the entire terrestrial planet, you can guarantee that the future of life is, the life I've described to you is definitely predictable. Extinction is predictable. it is going to happen around the world because we have so little land left, so little space left. And importantly, I guess, I get asked this question a lot, is can speciation rescue the biodiversity we already lost at the end of the Pleistocene, and that we're starting to lose now? We're basically poised on the edge of the sixth mass extinction. So is speciation gonna rescue it? And these, kind of all these base mutations that Tim described, those things take a while. You combine that with isolation of populations, and an average mammal, for example, takes between 2 and 5 million years to diverge into a couple of species. Some species are faster, birds tend to be a little bit faster, some species are much slower. In general, the point is that in our lifetime, there's no way that biodiversity can be rescued, in a sense, by evolution. We can't speed this rate up for us to see the difference in species richness on the planet by the year 2100. So to end with the question, is evolution predictable? I would kind of agree with Dmitri, but I'm also going to agree with Erin. There are parts of it we can absolutely predict. And the answer, to whether or not at the macro level evolution is predictable, we can say absolutely. We know when you have lots of area, you're likely to have more species if you give them time. It takes just a short period of time to eliminate diversity. All it takes is removing those individuals that harbor all of those millions to billions of years of genetic diversity they've included. That's what you carry around in your body, right? So it takes nothing to remove that, but it takes a long time to achieve it. So evolution is predictable in the sense that we know time makes a difference, that area makes a difference, and extinction, we will definitely recover in some way. Life will recover from the extinction event. But exactly whether or not it will be you or I or Stephen Jay Gould at Harvard, the answer is no. So with that, I leave it to Tim to lead the next part.
We have some time for questions.
And if there's questions from the audience, I ask the questioner to speak loudly so that everyone can hear. Professor Block, would you like a question?
Okay, this is loud. I think you made a good case. If it takes 5 million years to derive a species, man's been around for what, 2-3 million years? For 99.9% of that time, not a lot happened. The last 1,000 years, 0.1%, enormous technological change. We can now manipulate the genome and do other things. Have we reached the end of evolution? We can look back upon this as a very quaint period, and in the future, we'll be in a situation where evolution will be taken out of the hands of stochasticity and into our hands, and will we be manipulating the species of the future? And will we look back on this as a time when we allowed chance to reign rather than humans? or machines, for that matter.
So there's definitely a philosophical part of that question that I don't know, we could talk about that. But there is a lot of debate about, right now, I don't know if any of you have heard of the concept of rewilding. So rewilding is the idea of, well, if we can manipulate, if we can manipulate genomes, and if we could even resurrect species, passenger pigeon, heck, why not go back to the mammoth or the mastodon? What's stopping us from doing something like that? And why not? Why not just, basically recreate diversity? So, I have a really very strong opinion about that. And my opinion is we can't even take care of the species that we already have on the planet. We don't even have enough room for the species. There is a big extinction debt. And right now, there are species poised on the edge of extinction. We may lose the elephant in our lifetime, the African elephant. The mountain gorilla is history. Condors, history. So these animals are hanging on as individuals, but in terms of their persistence as, basically over the next century or 2 centuries or 3 centuries, we have a lot of extinction debt. And I would say rewilding is not the answer, because the answer is providing the place for them to live, not just having them as individuals.
So I have kind of a different answer to that question. That if we're so great at technology and engineering things, why do we have so many parasites and pathogens still that are really good at adapting to us?
This is not about our being great at it. It's a question
So-
about whether this is just going to happen or not.
Yeah, I mean, I think that despite our best efforts, evolution continues to foil our efforts in a lot of cases, especially with parasites and pathogens and things that we don't want to exist so much. Now, about the kind of diversity that we want, like what Liz is interested in, I think that's a little harder to predict. But I think, parasitism is a very strong lifestyle, and it's one that's very hard to overcome with technology.
I'm not sure we'd want one of those giant clawed sloths anyway. In the back.
About what?
During the age of the dinosaurs, we didn't just have megafauna. We had animals the size of this room, feasting on other animals the size of this room. And then after the Triassic, we reran the tape, and that didn't happen again. Do you folks understand why such large animals evolved and why they didn't evolve the second time we did the experiment?
Why is one of those questions that we're kind of trained not to answer as evolutionary biologists. So body size is a really important part. There's a lot we didn't talk about, obviously, but body size is really stunning. And there were in the Carboniferous, there were insects that were just gigantic, right? And so there's a sense, body size is an important. There's a lot of energetics behind it. There's a lot of thermal regulation behind it, and there's a lot of oxygen behind it as well. And so in terms of why we didn't evolve those giants, I don't know that I have the answer to that. We certainly have a diversity of other things, so we have lots of different ways of living than we did during the Triassic, at least apparently based on the data we have. Again, our diversity is a lot higher. And I mean, I guess that's how I'll leave it at that. Not a very satisfying answer, but.
And blue whale, I think, is the largest animal that ever
Indeed.
lived. So,
Yes.
Aren't populated areas just another kind of environment where new species can evolve?
So one of the kind of the trends in evolution that's also a little bit predictable in terms of the kinds of species that we're sorting. So there's a move to call the age that we're living now the Anthropocene. I don't know if you've heard this, but the Anthropocene has a characteristic suite of plants and animals, and they tend not to be things that like to kill us. So we tend to like to eliminate the big things or the things that are lethal. And we tend to kind of prefer things in our environment that are generalists, that are kind of weedy, that thrive in our disturbed environments, or that are somewhat, they're commensal, they're somewhat more domesticated. So one of the most wonderful examples that I've been working on in India is the difference between tigers and leopards. I mean, India is an incredible place. It's been occupied for probably 65,000 years. And what's amazing is they used to have cheetahs, lions, tigers, and leopards. So even Africa didn't have those four. And now there's about 115 Asian lions left. There are 3,000 tigers, and there are thousands and thousands of leopards. So why are leopards doing so well? Anybody know? So tigers, it turns out, only survive where people are not and where there aren't agricultural fields. And the reason is because they need deer. They need this intact habitat. They're very shy. They need cover. So they survive in generally remote, protected areas. Leopards, on the other hand, thrive at night. They're nocturnal, and they thrive on the margins of rural areas. And guess what they eat? They eat feral dogs. So there are, so many feral dogs in India, and leopards, it turns out, are just kind of coming out at night, and they're walking around people's houses, and they're thriving on feral dogs. So they're the survivor. They're both big cats They're both dangerous, but they're the survivor because they've learned how to shift their diet.
Won't we also expect that more species will evolve that will make use of this niche?
Coyotes.
Well, coyotes are not going to evolve, they're already evolved.
I think the time scale is.
Yeah.
So you need the behavioral plasticity, that's the generalist. You need a very plastic species in order to kind of co-opt that new resource. It's not really a question of independent evolution of a new species.
I have a question with respect to climate change. So we know that the Earth has gone through many ice ages, even in relatively recent history compared to some of the history we've talked about. Those are big changes in climate. And yet we are here today with, aside from human intervention, the very diverse set of species that we have. So as a provocative question, how much should we be concerned about this if Earth has already gone through potentially much larger changes over relatively recent history?
Sorry. So don't let anyone, tell you that the Earth has never been as warm as it's going to be in the next century, because that's not true. The Earth has been a lot warmer than it is right now. We weren't on it then. So the kind of changes we're going to head to, so the kind of projections for climate change, and this is where the fossil record really gives you some insights. So by the year 2050, temperatures will be warmer than we've experienced in our lifetime as a species on the planet. By the year 2100, temperatures will be warmer than virtually any mammal that presently exists on the planet has ever experienced in their lifetime. So temperatures by the year 2100, global average temperatures will be warmer than they've been in the last 14 million years. So to paleontologists, 14 million years is nothing, but when you start to think about all the plants and animals and humans that are adapted to these kinds of temperatures, we don't have those temperatures in our genome, at least not the recent past.
Yes.
I would like to know whether you think that we are going to evolve.
Well, we are evolving. Absolutely. If you think about, say, HIV epidemic in Africa, humans who are less likely to get infected are surviving, and those that getting HIV are not. And there's also a lot of evolution of sort of gene culture coevolution. So populations that have higher birth rates are expanding. Populations that are having lower rates are declining. And I think it's hubris to think that evolution ever stops. So it might really be a question, again, of timescales. I mean, what do you care about? What's the timescale in which you measure things? But we're continuing to evolve.
What kind of changes do you think we'll have?
So what kind of changes? I think we will become resistant to certain diseases. I mean, I'm very pessimistic, so you shouldn't. My wife who's sitting there just can't take it anymore. I think our population will go through a substantial crash, and whether or not we'll make through it is unclear. But again, I don't know when it will happen.
You're talking about population changes. I'm asking properties. Is our brain going to get bigger?
Well, our brain got smaller, right? So compared
From Neanderthals.
from Neanderthals. I think it's going to be some kind of a technological change. I think we will become, part human, part machine in some way. I think that's more likely to happen. I think culture.
We already are, folks. It's
Yeah.
already happened.
I think culture will dominate, and engineering will dominate in that sense.
Richard.
But there's some chance here too, right? So going back to India again, their fertility rates are amazing considering that what they eat is virtually nothing. So part of this is chance. Part of it is contingency. And so maybe, brain size is a big deal to us, but maybe survival and, producing tons of children is a different issue in developing countries. And the future really is in the hands of the populations that have the most individuals.
Richard?
So, Dmitri, you talked about how something complicated like the eye has evolved independently many times. So my question is,
special about this human intelligence that made it so hard to evolve that we haven't seen in other?
It's a great question. I often wonder whether it's true that it evolved once, and whether or not we have evidence that there was never. So one argument is that sentient life is self-limiting, right? So technology evolves and then it kills us. So it's unlikely it happened, but it's an interesting thing. It's an interesting thing to think about. But I don't know. I think that's one of those things that it's hard to think about events that happen once, if they really happen once. Do you have any?
Yes, Orange.
I think you can find examples of convergent evolution as many times as you want, right? You can say, okay, well, here's a marsupial cat, and here's a different descending cat, and look, they both evolved with the same as tigers. But that doesn't really tell me much, right? What I'm sort of asking, given a set of information, right, like given a cat, how long can I talk about the cat before I have no idea what I'm saying anymore, right? Like how long can I, what's the time scale before chaos takes over? Or conversely, if I'm trying to guide the evolution of a species into some nook that I want to, right, with all of my new CRISPR tools or whatever, how much do I need to know, right? Is the problem intractable? Is the problem something that I would only be able to solve in a lab, and then the moment that I set it out into the wild, the thing would run away from us? Or is there hope in that regard, right? I don't think that's clear to me.
One thing maybe one of you guys can mention is the history of the domestic dog and its morphological diversity and how long it's taken to achieve that morphological diversity.
Yeah, so I'm not sure it's quite, but I can talk. That's an easier question. So I'll answer that
I'm the
one.
moderator.
So the dogs, humans domesticated dogs. It's very interesting how it happened. It's not quite clear, but possibly, like the way Liz was talking about, we just tolerated wolves who were picking up scraps that were less aggressive towards us, and we kind of co-evolved with them. But the modern breeds were generated just in the last couple of hundred years, mostly by the British, and just great diversity of shapes and sizes. What's interesting is that if you think of the skulls of all of the dogs and you put them in some kind of amorphous space, imagining what it is, the space of skulls of dogs basically spans the space of all canids that ever existed. However, if you put that space and you say, okay, where's cat? The cat is there. Cat is not inside of that space. So it seems like there is some kind of something's constraining evolution of a dog's skull to be within that space. Now, maybe it's not truly constrained, so it'd be interesting to see whether it's possible to select a dog to look like a cat, which I think is a worthwhile enterprise, and I highly encourage you to try. Or a cat to look like a dog.
Of course.
But I think you have a point. So there is some constraint, there is some chance, and whether or not we can direct evolution in a particular way, it's not clear. But I think it's still not a useless question to ask whether something is predictable. Even if you cannot make it go here, you know it's going to go there. So the way Liz is talking, extinction is predictable, or that there will be predators and prey is predictable, which you can see, for example, when you look at fauna that evolves on islands. There's a single species arrives, and you eventually have all of the roles.
But all these dogs are the same general.
They're the same species.
That's right. They interbreed. So
That's
they're
right.
not there wouldn't be a different number on your scale.
So that's right. They're all the same. That's just a 1. No, dogs are really extraordinary. And actually, for those of you who are interested in cats, pretty much a cat is a cat. I mean, cats, big, small, cats always look the same. You do not have that same kind of morphological diversity in cat skulls as you do in dogs. Dogs are really extraordinary. I did want to point out something, and this is it's a little bit of a tough thing when you're a biologist, and you just throw out the word species. And as a paleontologist, we throw out the word species all the time, and we count the number of species. But remember, life is monophyletic. It's all connected. And in some ways, if you just take a single lineage, let's just pick on a cat. You take a single lineage of cat, and let's presume that it never bifurcates into multiple species, and it goes millions and millions of years, okay? The fate of all species is extinction. Eventually, that cat will go extinct. When I said to you the lifespan of a mammal, an average mammal, is between 2 and 5 million years, we don't have many mammals that are older than 5 million years. So in a way, the fate of species is also divergence. It's divergence or extinction. And it's arbitrary where we draw the lines between what we call paleospecies or even what we call modern species. And that's why I kind of threw out viruses and bacteria. And so species itself is one of those concepts that's constantly debated in evolution. And it's really important because you can get pretty circular pretty fast.
I think on that note, the diverge or perish, which is kind of like the publish or perish in academia dictum, we're going to call the evening to a close. I thank you all for coming, and I thank our panelists for a great discussion.